Good morning, everyone from Colorado. Welcome to the first COMMeND webinar of the year. So happy New Year to all. And we are seeing participants from different places around the world. Thank you for joining in. If you have questions about COMMeND, you are free to browse through our website or contact Landon Mayer. We can put in his email later in the chat. So some ground rules. We have two great speakers today talking about biofilms for otitis media, and we would recommend that you put your questions in the chat. We will also try to have an interactive session after the second talk before we move on to the session with Rita Lee on effective communication. To introduce our first speaker, I'm really proud to have her in our COMMeND webinar. Professor Lauren Bakaletz is from Pediatrics and Otolaryngology at Ohio State University College of Medicine and Nationwide Children's Hospital in Columbus, Ohio. She serves as director of the Center for Microbial Pathogenesis within the Abigail Wexner Research Institute and serves as vice president of basic sciences, holds an endowed chair in pediatric research, and is named Distinguished Professor in 2013. She has been elected to the American Academy of Microbiology, and she is also an Allen Distinguished Scholar in Pediatric Research, as well as a fellow in the American Association for the Advancement of Science. She has been funded by the NIH and industry for nearly 30 years, including in molecular mechanisms that underlie chronic and recurrent polymicrobial infections, wherein bacterial biofilms contribute significantly. This includes otitis media, chronic rhinosinusitis, COPD, and cystic fibrosis. She has an ongoing collaboration with Dr. Stephen Goodman regarding eDNA and bacterial DNA B2 proteins. I think she will be talking about these today. And she's also a founder with Dr. Goodman on two startup companies conducting first-in-human clinical trials, conducting these on community-acquired bacterial pneumonia in people with cystic fibrosis. Floor is yours, Lauren. We're so happy to have you. Thank you so much.
Thank you, Reggie, and I appreciate that kind introduction. So I'm going to be the first of two speakers. As you know, Dr. Garth Ehrlich will also be joining in this webinar, but I'm going to start, and I want to begin by showing my disclosures. Can everybody see my slides and hear me? Is everybody good? Excellent. Okay. So I might be preaching to the choir, and no, I do, for many of you when I talk about bacterial biofilms, but it's really important to understand the role that biofilms play in the pathogenesis of chronic and recurrent diseases. And here on the left of my screen is the classic model put forth by Dr. Bill Costerton, who's really the grandfather of this field, and unfortunately he has passed away. But bacteria in nature prefer to live in a biofilm, so they will go and they will sit down on a surface and become irreversibly attached. Those biofilms will mature. The bacteria will produce an extrapolymeric substance matrix in which they reside. They form these microcolonies, although they are not all mushroom-shaped. They have a variety of different architectures. But these bacteria can disperse themselves from biofilms when they're under stress, when nutrition is limited, or when there's antibiotics or an immune stressor. Importantly, and in this image now that was put forth by Karen Sauer in a recent paper, we've come to understand that biofilms do not have to be attached. They can be aggregate biofilms, which are free-floating. And very commonly, this would be the type of biofilm you would find in the lung. And it is a form of biofilms where these organisms have all the same qualities of other biofilms that are attached. So bacteria in a biofilm, as I mentioned, are encased in a self-produced protective EPS, or extrapolymeric substance. This confers a lot of protection to them and allows them to reside in a body site for a very long period of time. Those biofilms don't have to be pathogenic. There are, of course, very good biofilms that we live in partnership with, but when they are pathogenic, they are particularly problematic. Bacteria within a biofilm are metabolically quiescent. This can be problematic because it makes them resistant to antibiotics that perhaps might target bacterial cell division or cell wall synthesis to be effective. PMNs, or white blood cells, and macrophages do have limited access to bacteria within a biofilm. And these bacteria, as I mentioned, can disperse themselves when they're under stress and go forth, leave the biofilm, go to another site, sit down, and start the process all over again. It's really important to understand that this is the preferred way bacteria like to live in nature, whether they're in the body, on an inanimate surface or an animate surface. Now, biofilm diseases are very common. They're complex. They're very recalcitrant to treatment. But 2/3 of all bacterial infectious diseases involve biofilms with the latest CDC estimates. And these can occur anywhere in the body. Biofilms very much like to sit down on devices that have been implanted in the body. And virtually any body site can manifest a pathogenic biofilm disease. Now, I'm going to be talking about otitis media today as an example, and many of you know I've worked in this area for the majority of my career. I have started to branch out my evil empire, but I hope to convince you that a lot of the principles I'm going to be talking about today that confer such resistance to biofilms are those principles that can be applied to them regardless of where they are in the body. Now, I'm going to start with showing you a very old image we have of a biofilm formed in the middle ear by non-typable Haemophilus influenza. Many of you know this as a predominant gram-negative bacterial pathogen of otitis media. We model in the chinchilla, as does Kevin Mason and Garth Ehrlich and others in the field. Here's a healthy chinchilla middle ear, and here you can see the tympanic membrane. It's just the lower aspect of that membrane. And within the middle ear of a chinchilla, which has the consistency of about a ping pong ball, you can see that it's a quiet mucosa. It's a single cell of epithelial cells that line the middle ear mucosa. But there are these interesting natural bony septae within the middle ear of a chinchilla that we like to use as depth gauges to try to estimate how much biofilm is in that middle ear. Now, if you put only about 500 non-typable Haemophilus into the middle ear of a chinchilla, about 4 days later, you'll see that that entire middle ear is filled with biofilm. These bony septae are obscured. This ear would rank a 4+ on our scale, which means they're 100% filled with biofilm. Quarter filled would be a score of 1, half would be 2, 75% would be 3, and 4 is 100%. And you'll see this come up later in some of my data. Notice that the eardrum is very red and inflamed, and very similar to what a pediatrician is likely to see when they look down the auditory canal of a child who has otitis media. Now, if you take this biofilm out of this site, you snap freeze it over liquid nitrogen, and you impart a live dead stain where the live bacteria are green and the dead bacteria are red. You can hopefully appreciate that virtually all of these bacteria are indeed alive. These long, thin, green finger-like projections that extend up from the middle ear mucosa into the middle ear lumen are towering communities of millions of bacterial cells, all clearly very, very viable. These biofilms in the middle ear, as I've mentioned earlier, are resistant to killing by innate and acquired immune effectors, highly recalcitrant to antibiotics. The estimates are that there are 1,000-fold greater doses are needed, but I would have to say, in my experience, it can far exceed that in terms of their ability to resist antibiotics that readily kill them if they were growing planktonically or in liquid medium. And they do play a primary role in the pathogenesis, recurrence, and chronicity of otitis media. Now, many years ago, we decided, could we figure out what these biofilm resident bacteria were doing that might be unique, that we could then target to see if we could either prevent them from forming a biofilm or disrupting them if they were already in a biofilm. And I can tell you that we honed in on extracellular DNA, or eDNA. Now, within otitis media, in the middle ear of chinchillas and children, there's an abundance of extracellular DNA. We were not the first to discover this. Cynthia Witchers did in a really land-breaking paper in Science many years ago. But the DNA in a biofilm comes from two sources: the white blood cells that net and extrude their own eukaryotic DNA around the biofilm to help cordon it off. But within the biofilm itself, it is pure prokaryotic or bacterial DNA. Now, what intrigued us was that this DNA, whether PMN origin or bacterial origin, appeared in this lattice-like structure. And this was really intriguing to us. And we wondered what was mediating the weaving of this bacterial DNA into this lattice-like structure. And I had the opportunity to meet Steve Goodman at the time. He had been studying this group of two little bacterial proteins called the DNAB2 proteins, IHF, which is integration host factor, and HU, histone-like protein, they bind to DNA. So they have this little lobster shape. They will bind to unbent DNA and bend it. They like to bind to pre-bent DNA because it takes less energy to bend it. And what they really love to bend to or bind to are shapes that look like this, like holiday junctions. And these are transcriptional intermediates. Now, DNAB2 proteins are known for their roles inside a bacterial cell. They're involved in a whole host of nucleoprotein interactions. But these shapes reminded us of what this biofilm eDNA looked like. And we wondered, were the DNAB2 proteins not only inside the bacterial cell and doing all their important nucleoprotein interactions, but were they also outside the bacterial cell and perhaps responsible for weaving this lattice of DNA or weaving this bacterial DNA into this lattice-like structure? So Steve Goodman gave me some rabbit antibody to E. coli IHF. This is a non-typable homophilous biofilm from a middle layer of a chinchilla. In white is the eDNA lattice. So the eDNA strands of double-stranded DNA are labeled in white. And I hope you can discern that every cross junction, every vertex of cross strands of eDNA, we got specific and very distinct labeling with this antibody, suggesting that not only were these DNAB2 proteins also operational outside the cell, but they were probably providing essential structural support to that protective biofilm matrix. So being the daughter of an architect, I wondered if we could pull these out of this lattice. Would this collapse the biofilm? But first, reviewers wanted to know, was this observation unique to just homophilous biofilms in the middle layer of a chinchilla, or was it perhaps more broadly relevant? And I can tell you, and here's just a sampling of samples we've looked at. This is otorrhea from a child with the tympanostomy tube. You can see the lattice of eDNA and the abundant labeling of the DNAB2 proteins at the junctions of these cross strands. This is an adult with sinusitis. This is an intracellular biofilm caused by uropathogenic E. coli within a mouse bladder. And this, unfortunately, is sputum from a patient here at our hospital with cystic fibrosis who's also culture positive for Burkholderia sinusopatia. That organism produces an abundant amount of eDNA and DNAB2 proteins. And you can see that the eDNA in this is almost obscured by the amount of DNAB2 protein within that lattice of bacterial DNA. So back to my question was, what happens if we remove these proteins from the lattice? Our hypothesis was that if we remove them from the biofilm, this would allow collapse of the eDNA scaffold and release of the resident bacteria. And we approached this in a number of ways. We used a very classic in vitro biofilm disruption assay with a covered chamber glass, both looking at otopathogens, but also other high priority pathogens. We looked at multiple polymicrobial clinical specimens to see if this observation held. And we evaluated the efficacy of this approach in 3 unique animal models of human disease: the chinchilla model of NTHI-induced otitis media, a murine model of Pseudomonas aeruginosa-induced lung disease, and also a rat model of periimplantitis due to Aggregatibacter actinomycetum concomitans. And I'm going to condense for you in one slide about 12 years over, which is sad or good, just depending on how you look at it. Anyway, the biofilm extracellular polymeric substance contained an abundance of extracellular DNA in every specimen we looked at. And importantly, the bacterial DNAB2 proteins, we were able to detect them at the key structural junctions within the DNA of many bacterial species. And here are some of the papers we published that documented that. But what was important and what was interesting to us was that antibody directed against the DNAB2 proteins did indeed induce collapse of the biofilms. So here you're looking at a homophilous biofilm from the top, growing in a chamber slide. And these side views are what would be the orthogonal views of those biofilms to give you an idea of biofilm height. When we allowed homophilous to form a biofilm, and this is a live dead stain, you can see it's clearly very alive. And we incubated that biofilm with naive rabbit serum virtually nothing happened to the biofilm. But when we incubated it with a random 1 to 50 dilution of antibody directed against one of the DNAB2 proteins from Haemophilus, you can see that this biofilm collapsed precipitously. The more antibody we use, the greater collapse. Now, this antibody doesn't kill the bacteria. It simply collapses the biofilm matrix and releases the bacteria. And you can take these biofilms down to a monolayer. So in answering the question of, was this an observation unique to just Haemophilus influenza, as I intimated in the previous slide, we've been able to disrupt biofilms formed by all of these diverse human pathogens, not only singly, but also mixed specimens from cystic fibrosis, individuals with cystic fibrosis, otorrhea exudate, cesarean section wound infection, which are very much polymicrobial, and importantly, all of the high priority escapee pathogens. These are the pathogens that the World Health Organization is most keyed in on because of their incredible multiple antibiotic resistance. Now, it was great to see the disruption of these biofilms in vitro, but what was really compelling to us was that in all three of these animal models that I mentioned, as well as a new one, we have a new murine model of non-tuberculous mycobacterium infection of the lung. That manuscript is in revision. We were able to not only disrupt the biofilms and release the bacteria from these infected sites in these animals, but with absolutely no delivery of antibiotics. We had resolution of disease and return of the mucosa to homeostatic conditions. So that was really, really intriguing to us, but also important because antimicrobial resistance is really considered yet another global pandemic at this time. So the question that we got asked, and it's a really important one that I like to think about as someone who develops vaccines and therapeutics, is if the DNAB2 proteins are sitting there in these biofilms, why don't we mount an immune response to it and disrupt these biofilms on our own volition? Why aren't we developing an immune response to it? And our hypothesis was that perhaps when the DNAB2 proteins were integrated into that DNA within the biofilm, their protective epitopes were masked because they were intercalated into that eDNA. So if I were to show you a three-dimensional map, this is a native IHF protein. And like I said, it looks kind of like a lobster tail. So here's the tail and here's the two arms. Native IHF versus IHF that's already been pre-bound to DNA were the forms we used to immunize chinchillas. So we could get antibody to the form that's found in a biofilm or antibody to the form that is the native form of the protein. And what we found was that this antibody, and we got a robust response to it, had absolutely no effect on disrupting the biofilm. But antibody to the native protein was able to disrupt the biofilm. So when we epitope map this immune response using a series of 20-mer peptides with a 10-mer overlap, what we were able to find was that what happens in the body during a pathogenic biofilm is that the immune response is really only recognizing this tail of the IHF protein or DNAB2 protein, versus when it's in its native state, you now start to see a shift to response to the tips. And I'll rotate those for you so you can see that a little bit better. So it was very, very clear to us that our hypothesis seemed to be right. This form is immunogenic, but the immune response is misdirected. It's directed to the tail, which has no effect on disrupting a biofilm. So we set out to design a better DNAB2 derived immunogen, not only to use as a vaccine candidate, perhaps, but to also use as the model to which we would make the monoclonal therapeutic that we are currently testing in human trials. And so this time we were focused specifically on those protective domains. Now, if I show you this map again, I can tell you that at least IHF is a heterodimer. There's an alpha subunit here in green and a beta subunit here in orange. We knew from our mapping studies that these tail proteins were not protective. All of the immunoprotective domain was in the tips of the proteins. So we designed a peptide immunogen that included a 20-mer region from the alpha subunit tip, a small linker peptide to give flexibility, but not let these two wrap around each other and interfere with the immune response, and a 20-mer from the beta subunit tip. Now, this is called a tip chimer peptide. It's a 44-mer. But to use as a control, I also designed a tail chimer peptide using 20-mer amino acids from the tail of the alpha subunit, 20 amino acids from the tail of beta subunit with the same linker. And this is called the tail chimer peptide. This is intended to be the positive control, and this is intended to be the negative control. So what happened when we immunized animals with it? Now, the question we had at the time was, well, why do you need a better immunogen? Why don't you just immunize with the native protein? The native protein induced antibody that disrupted the biofilm. So why are you going to this effort? Well, I'll show you why. So on day 0, we inoculated chinchillas right into the bulla with Haemophilus to induce otitis media. We waited 4 days because we know that that ear will be filled with biofilm in those 4 days. And then we delivered immunoglobulin IgG directly into the middle ear. This is about 5 micrograms. So we used either naive rabbit serum. We used rabbit serum to the tail chimer. That's the negative control. We used rabbit serum to the native protein. And we used rabbit serum to the tip chimer, the positive control. 1 day later, we determined the bacterial load in the middle ear. We brought about 15 people into the room and asked them to blindly assess how much biofilm was left in that ear, assuming that at day 4 it was 100% filled. We also then had another subcohort that we let go another week because people said, well, we can still see, what if we still see biofilm here? Your antibody doesn't really kill the biofilm. So what if those few bacteria left in the monolayer regrow? Are we going to see return of disease at day 13? So what I can tell you is that 1 day after delivery of 2 doses of antibody, this is the naive serum. We had a load of about 5 times 10 to the 6th Haemophilus in the middle ear. When we delivered the tail chimer serum here in blue, there was no diminution of bacterial count in the middle ear, but either the anti-tip chimer peptide in yellow or the anti-native protein reduced this load of bacteria in the middle ear significantly. There was at least about 4 log reduction in counts. But at this point, we really couldn't say the tip chimer was better than the native protein. When we asked blinded observers to tell us how much biofilm was left, you can see the middle ears of the animals that got the naive serum or the tail chimer serum were 75 to 100% filled with biofilm. But the biofilms in the ears of the animals that got the anti-native protein were sitting here just above 1, and we're starting to now detect some superiority in terms of the reduction in biofilm by delivery of the tip chimer peptide. However, whereas these two were significantly different from the negative controls, they were not significantly different from each other. So we then looked at what was the durability of the biofilm eradication 1 week after therapy was completed. To give you a frame of reference, here again is a healthy middle ear that would score a 0. Here's the middle ear filled with biofilm that would score a 4. And what we saw with naive serum was that it looked just like animals that had no treatment at all. This was a 4. Anti-tail chimer was not only filled with biofilm, but it had really induced an inflammatory response in those middle ears. And that's a whole different seminar. And I could tell you why if you're interested, and we can talk offline. But in both the animals that got the native serum, anti-native serum, or the anti-tip chimer, we really had a hard time detecting much biofilm, if any, in the middle ears of these animals. If there was biofilm, it tended to cling to these bony septae. And you can see the capillary bed in the inferior aspect of these middle ears, which tells you that that mucosa was no longer hyperinflamed and thickened. You're still able to see that capillary bed, which was intriguing to us. So could we develop an assay to tell us for sure which was better, the native protein or the tip chimer? So we developed an in vitro assay where we allowed biofilms to grow to a biomass, a pretty large biomass of about 11. And we then treated them over a course of 3 hours with either IgG from naive rabbits or IgG from animals given who had been immunized with the negative tail chimer peptide. And as you can see, they had no effect on that biofilm over those 3 hours. When we gave these animals DNase, like palmizyme, as we use in people with CF, there was a little bit of an effect on the biofilm, which is surprising, right? Because biofilms are so filled with DNA, and DNA nucleases should digest the DNA, and I'll tell you why they're not able to work. It was a little bit of an effect, but nothing impressive. Look at how rapidly IgG from animals immunized with the native protein were able to significantly disrupt these biofilms and hold those for that period of time. But now we're starting to see that with the IgG from the tip chimer serum, we had a markedly better effect and was significantly different even from the anti-native IgG. So we knew that there was superiority, but was that still enough to warrant going into humans or to move forward to clinical trials? And this was the question: do we need a better immunogen? And to get to the bottom of this, we want to assess how most of us walking around with either a history of having biofilm infections, or particularly kids with otitis media, how were they recognizing the DNAB2 proteins, which had to be within the biofilms in their middle ears? And what we found was that in all of these children, these were archived pediatric sera, and these are pediatric sera we got from kids having their tubes inserted in the time of frame we were publishing or trying to publish these data. You can see that their response uniformly was greater to the tail of that protein, because the tail is what's available when these proteins are in a biofilm. There is also a response to the tip, but it is less, hierarchically less than that to the tail. And I will point out for you, there was this one kid who was the opposite. There's always one, I don't know why, but I left him in, which is almost an even response to tip and tail. Now, in healthy adults walking around in my center, again, really predominant response to the tail. The tail was greater reactivity in 94% of archived pediatric sera, 100% of fresh pediatric sera, and 100% of fresh sera from adults, which evoked concern in my mind for original antigenic sin. Now, for those of you who don't think about original antigenic sin, this is the propensity of our body's immune system to preferentially use memory to respond to a second infection based on what it saw in a first infection of a similar entity. So this is why we need a new flu shot every year. Otherwise, our body would keep responding to the flu virus it saw last year and not be as effective as the variety we have this year. And so what we saw was we took 2 chinchillas, and chinchillas also have a predominant tail response, a diminished tip response. When we immunized the chinchilla with the native protein, once, twice, or 3 times, we preferentially drove the response to the ineffective tail. This would not be a protective. In the animal that we immunized with the tip chimer peptide, not only did we hold the response to the tail stable, we selectively induced a tremendous response to the immunoprotective tip. So our strategy is that what we need to do is redirect the natural immune response to the domains of those proteins that are indeed protective. So where we are with this now, after about 15 years, is this is what we think is happening. So here's Haemophilus, these little green bacteria. They put their own DNA out into the biofilm. They lock it in place with these DNAB2 proteins. The majority of those are in the biofilm, but there are a few out in the extracellular milieu. If you add antibodies specific to the tips of those proteins, it will bind the DNAB2 proteins that are free, induce an equilibrium shift so the other DNAB2 proteins come off the biofilm collapses and releases these bacteria in a form we're referring to as newly released, or NREL. This is fast, specific, dose-dependent. You do not need direct contact between the antibody and the biofilm to induce it. But importantly, really importantly, these newly released bacteria, there's a window of time where they are highly, highly sensitive to killing by both innate immune effectors, acquired immune effectors, and antibiotics. So this is something that we are really trying to bank on in our human trials. But this brings me to the question of why, if bacterial biofilms are so rich in DNA, can we not just treat these diseases and these individuals with things like pulmozyme or DNase? And that brings me to the subject of Z. Now, probably you, like I, or maybe you're better than me, but when I first learned about zDNA, I promptly forgot about it because I've always been focused on bDNA, your classic, right-handed helix, a Watson-Crick base pairing, etc. But DNA can also exist in other forms, the A form, the B form, the Z form, and those are shown graphically here. But what's really important is the Z form of DNA is left-handed, it's nuclease resistant, and it's in such a high energy state because it's so unstable that it had never been observed extracellularly in a stable state until recently. And probably the best way you can think about zDNA is if you took a rubber band, held it with both ends, and just start twisting it and twisting it and twisting it until it twisted up on itself. You would find this thick, ropey, entwined molecule, and that is zDNA. When we looked at biofilms, and these are biofilms this is Haemophilus, these 4 panels, this is uropathogenic E. coli, this is Streptococcus mutans, and this is Klebsiella pneumoniae. These are 40-hour biofilms. We stain them, so they're stained for this is FM464, which is just for the bacteria themselves. You can see the biofilm, and here's the biofilm stained with B and Z DNA. Here's the biofilm stained with a control antibody. But when we remove this signal from FM464, you can see that there's both blue and yellow. So there's both blue bDNA and yellow zDNA in Haemophilus biofilms, UPEC biofilms, S. mutans biofilms, and Klebsiella biofilms. They're all there. So does DNA selectively, so the question was, well, if you treat these biofilms with DNase, it should get rid of the bDNA and leave just the zDNA. And that is indeed exactly what we saw. So when we have either UPEC biofilms, Haemophilus biofilms, Klebsiella pneumoniae biofilms, these biofilms are a mix of B and Z DNA. When you treat them with DNase, the bDNA largely disappears, and you can see that graphically here, but the zDNA remains. And notice that we do not have a loss of biofilm size when we remove the zDNA. So the biofilms stay intact, they stay architecturally stable, even though the bDNA is gone, because that zDNA is imparting that structural stability. So what would happen, let's say, if we shifted all of this DNA from B to Z? And we can do that by treating biofilms with cerium chloride. So cerium chloride binds the phosphate backbone of DNA, neutralizes its charge, and allows the zDNA to be favored. So here's a biofilm with antibody control. This is a Haemophilus biofilm. Here's a biofilm labeled for zDNA without cerium chloride. And now we've shifted that biofilm bDNA to zDNA. You can see the tremendous labeling of zDNA. And I'm not going to go through all of these in the interest of time, but what I can tell you is that the fluorescence intensity of the zDNA goes up. The fluorescence intensity of the cells go up. So when you do this, it tends to drive more bacteria into the biofilm than are in the planktonic state. And importantly, when you put a stressor onto these biofilms and you're trying to assay for rheological properties, these biofilms are larger and they're stiffer. They are much more rigid. And so these biofilms are energetically favored to not be disrupted by anything that we're doing to them. So these biofilms are becoming larger and more rigid. Are the DNAB2 proteins, though, responsible for this? And we do know now, and we published this recently in Cell, that when you add DNAB2 proteins, so here's a biofilm without a DNAB2 protein, it's labeled for just the bacterial biofilm. When you add HU, which is a DNAB2 protein, you can see that you do increase the zDNA content by just adding the DNAB2 protein. So what the DNAB2 proteins are doing is, I told you, they sit at those junctions of eDNA and they just twist it, and they twist it, and they twist it, and they twist it from B form to Z form. And that's making it thick, ropey, and nuclease resistant. And it is the DNAB2 proteins that are responsible for this conversion. So the question was, though, we know, Dr. Goodman knows that once the DNAB2 proteins switch the DNA from B to Z, they can leave the biofilm. So what do? Well, we wondered, what if they're migrating from the biofilm to go to the B DNA from the PMNs, the white blood cells, and can they convert the PMN DNA that's in the net that's designed to capture and trap and kill the bacteria, and inactivate that function? And that is unfortunately exactly what we saw. So this is a PMNs incubated with just some stains to label the PMNs. We have wheat germ agglutinin for the PMNs, the neutrophils without HU, which is a DNAB2 protein. When we induce those PMNs to net, they do put out all their own bDNA. They're making this big net of bDNA to capture the bacterial pathogens. When we incubate them with HU also, you can see that we start to see a loss of the blue signal. What we do see is an increase of the yellow signal. So with HU, the bDNA from the PMNs is also being converted to Z. Well, why is that important? Because the antimicrobial properties or the antimicrobial products that bind to the nets of PMNs that kill bacteria cannot bind to the Z form of DNA. So they are released from it. And not only that, but the PMNs, which are readily able to kill bacteria when you incubate them with, so here's bacteria alone, bacteria with neutrophils are killed by the neutrophils. If you add DNase to get rid of the bDNA, you lose the ability of the PMNs to kill the bacteria. Same thing when you add HU. This is a new DNA binding protein that Haemophilus produces. It has no effect on the B to Z conversion. And so this is not disabling, but the DNAB2 proteins are very disabling to the PMN killing function. So could we demonstrate that this occurs in human or in disease? And I'll very quickly go through this, hopefully for you. This is mucosa from the middle ear of a chinchilla, and this is the lumen of the chinchilla here. There's a tremendous amount of DNA, and this is just DAPI labeling. But what you can hopefully see here is that here's the mucosa, here's the biofilm. DAPI does not label zDNA. This entire wall of blue labeling is DNA from PMNs. So when we label the same biofilm for zDNA, you can see that the biofilm itself is zDNA, but there's also zDNA throughout this mass that we recovered from the mid-ear. Notice that the closer you are to the biofilm, the lack of signaling from bDNA. So the DNAB2 proteins from that biofilm, we're positing, have migrated from the biofilm, flipped the PMN DNA from B to Z, and you have a mix all the way through the biofilm. But if you look at it from bottom to top, biofilm alone is Z, pure Z. In this region closest to the biofilm, here's all the dead PMNs, those are the gray bodies. All that DNA is Z. As you move up, you get a mixture, but the only place we see pure B DNA is with the PMNs that have just migrated into this middle ear. They're sitting on top of this wall of dead PMNs, and none of them are affecting this biofilm. It's not eradicated in a chinchilla. Do we see this in humans? Yes. So these are sinus mucosa samples recovered from children with chronic rhinosinusitis here at Children's Hospital. Mucosa is at the bottom. Now zDNA is green, bDNA is pink or fuchsia. So all this wall of zDNA in the biofilm sitting on the mucosa from these sinus cavities of these kids. The only place we see B is in the most recently migrated PMNs. And you can see a couple of them have gotten into the biofilm, but they clearly haven't been able to eradicate it. So what we think is happening, and this is my last slide, is that you've got these bacteria, now gray, in a biofilm, and they're putting out their DNA. They're locking it into the lattice with the DNAB2 proteins. And there's, again, some free in the medium. When you add nucleases, which should cut the DNA and cause the biofilm to collapse, allowing the PMNs to come in and put out their bDNA, releasing histones and killing the released bacteria, that is what we hope happens. It doesn't, however. And we don't get this clearance of the biofilm and the killing of the bacteria that are residing. Instead, what's happening is that this DNA is being converted from B to Z form. So once the DNAB2 proteins twist this DNA into its Z form, it's not only nuclease resistant, but insidiously, these DNAB2 proteins can migrate from the biofilm to the bDNA released by the PMNs in an attempt to wall off the biofilm and kill the bacteria, converting it to Z form. So they are no longer effective in that functionality. So to wrap up for you quickly, we do believe that eDNA and DNAB2 proteins are really integral to the structural integrity of diverse pathogenic biofilms. They do induce the conversion of B to Z that not only fortifies the biofilm matrix, but inactivates a really important defensive function of PMNs and makes these biofilms nuclease resistant. If you remove them with specifically epitope-targeted antibodies, you can collapse the biofilm and release the biofilm resident bacteria. These newly released bacteria are highly, highly vulnerable. We've published a number of papers recently. We have a couple other out there in revision that are talking about those qualities of those newly released bacteria and why they're so vulnerable. But importantly, we think that this provides a window of clinical opportunity so that if you could treat people with biofilm diseases like kids with otitis media with this antibody, collapse the biofilm, release these newly released bacteria that are highly, highly vulnerable. You can kill them either with the normal immune response of those kids or by co-delivered antibiotics. And I hope that this theory is right. We're currently in 2 clinical trials, first in human clinical trials for people with bacteria, community-acquired bacterial pneumonia, and we're also doing a trial in people with cystic fibrosis. And we expect results hopefully a little bit later on this year. And so please keep your fingers crossed that this is effective and helpful for these individuals. So a lot of people to thank, both past and present in my own lab and Steve Goodman's lab, in the lab of my colleague, Dr. Santiago Partida Sanchez, the C3 immune cores that provided us with sputum from people with CF. All those individuals in yellow are the ones who generate the majority of the data, as well as those highlighted with the yellow asterisks. Also, very, very grateful to the late, great Paul Studley, a wonderful, wonderful person who is very much integral to the biofilm community that we lost late last year. Aaron Globe, who used to be his PhD student, who is now faculty at Virginia Tech. And of course, we are always, always grateful to NIDCD for funding. They have funded me my entire career, and I am eternally grateful. So I'd be happy to answer any questions, and thank you for your attention.
Wow, Lauren, just wow. That's so excellent. I've heard you talk about biofilms before, but every time I hear you, you have something exciting and new. So thank you so much. I'm going to take host privileges because nobody put anything in the chat. I think they were really into, oh, there. Stephen has his hand up. Maybe we'll have Stephen's question first. Go ahead, Stephen.
Hi, that was a wonderful talk.
Thank you.
So this is really interesting for me as well. So I'm doing a lot of otitis media in the context of strep pneumo, not Haemophilus influenza infections. And recently our lab, we're really looking at neutrophil functionality, but we decided to take a look at potential outside phagocytosis, because phagocytosis is important for strep pneumo killing. But because we had read some papers showing that there was a lot of nets in clinical otitis media, we wanted to look at the role of PAD4 mice. And I just want to know, like, just a bit more, because our PAD4 mice, when we try to induce otitis media, have significantly lower CFUs and bacterial burden for strep pneumo. And so PAD4 is involved in the pathway to create nets. And so, like, with just your talk, is just getting me just thinking in general that, like, this pathway is helping the bacteria just create an extra protective layer with their biofilm with Haemophilus. And like, just, do you know of the role? So for killing Haemophilus, to really get to the main point, is it an extracellular mechanism of killing that neutrophils do, or is it more of this intracellular, or is it either or?
It's actually both. And we do know that the intracellular killing is done in an NADPH oxidase sensitive manner, because we can inhibit that killing of the NRO. We can inhibit that much enhanced killing of those newly released bacteria if we treat the PMNs that way. But they're also very, very rapidly killed extracellularly. And we have a paper that we're about to submit right now, which really shows that PMNs, when they're exposed to these newly released bacteria become hyperactivated. They release a tremendous amount of IL-8. They migrate rapidly into the site, and they both phagocytize and they net. So you can measure a huge difference, a significant difference in both phagocytic activity, but also netosis activity. So there's something unique that the PMNs are detecting that's not difference in growth phase, not endotoxin, not mediated by strain. It's a common phenomenon that we've seen. So I think that story is one that you're very more likely going to be putting some chapters into, because I am not a PMN person. I kind of stumbled on this in pursuit of some of these other therapeutic and preventative strategies, but there's a definite interesting story there. What was amazing to me was how many PMNs had come into the middle layer of that chinchilla. There's layer upon layer upon layer upon layer, many, 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 many layers, completely ineffective.
Yeah, we get when we look via flow cytometry, we've looked at 6, 12, 12, like we have a very robust amount of neutrophils coming in.
Yeah.
But for at least for strep pneumo, like phagocytosis is very important for killing that.
Right.
But we don't see the same killing. And then when we block PAT4, which, I mean, this is a difference between different diseases, at least. Like it really helped out, as well as other factors we've done to try to increase neutrophil killing activity. But that's just really interesting to how, like, with your talk and the overall story, that this function that is normally killing for Haemophilus is just the bacteria just completely disrupts it, and then you're creating this just additional protective layer of dead neutrophils that you then cannot get to the back bugs at all.
I mean,
so it's just really fascinating for me.
Think of it from the bacterial standpoint, not so clever from the kid's standpoint, but from the standpoint of the organism, and it's incredibly effective, right?
Yes.
It's not only defensive, it's offensive.
Mm-hmm. No, and it's just the talk is just so relevant because of just the direction we just recently went in to look at nets.
Thank you.
Thank you.
Good luck.
Okay, well, Mary has a question. She can unmute to ask her question.
Hi, Dr. Bakaletz. That was an amazing talk. Kind of similar to your like defensive questions. I was wondering, I guess, knowing that bacteria can produce nucleases in order to like compete with other bacteria, have you done any like competition assays in a biofilm to see if maybe the production of zDNA could be used as like a defense mechanism for bacteria?
We haven't, but I would imagine that it can be. I think it's not only protective and it can be competition, but I do think that's how many bacteria release themselves from a biofilm, right? They make their own nucleases, and I think probably what happens, they downregulate DNA B2 release to get that Z flipped back to B so that they can then digest it with their own nucleases. And in fact, that's another therapeutic strategy we're working on right now, because chloroquine, for example, it's been used for malaria. Can flip Z back to B. And so we're working on, is there a way to maybe combine these approaches to really hit these biofilms in multiple different ways to be effective? So it was a super fun discovery because it was so unexpected. When you stumble on it, it's like, nah, this can't be real. But I think I'm more and more convinced that this is a really important way that Haemophilus and other bacteria do defend themselves in a biofilm. And that is why we can prevent biofilms from forming with DNase. But once they're mature, there's been no ability of anybody to really do anything significant with nucleases and biofilms of any formed by any pathogen or clinically even. Thank you, Mary.
Thank you.
Yeah.
I don't see any new questions or hand raised, so I'll proceed with introducing Garth. Thanks again, Lauren. We'll save some of your other questions later for interaction or discussion.
Okay.
All right. So our next speaker is Dr. Garth Ehrlich, who is a professor of microbiology and immunology and also otolaryngology, head and neck surgery at Drexel University College of Medicine in Philadelphia. His scientific career has been characterized by his integration of technologies and ideas across a broad spectrum of academic and clinical disciplines. And that includes diagnostics, informatics, AI, infectious diseases, genetics, wound healing, and evolutionary biology. He was elected as a fellow of the American Association for the Advancement of Science and the American Academy of Microbiology. And he was recently chosen as a member of the American Society of Microbiology's Distinguished Lecture Panel. His major contributions includes rewriting much of our understanding of chronic bacterial pathogenesis. He is really a giant in the field, and he began with his promulgation of biofilm paradigm to explain many facets of chronic mucosal microbial infections. In the interest of time, I'm not going to read his entire blurb, but maybe we can post this when we post the same with Lauren when we post the webinar online. But his latest work characterizes insight to evolution of virulence among bacterial pathogens during chronic infections and the development of high-resolution microbiome analysis, such as species-specific pan-domain MDX assays and novel antibiofilm drugs targeting bacterial stringent response. So we are excited to hear Garth talk. Please go ahead, Garth. Thank you.
Reggie, thanks for that very kind introduction. And Lauren, thanks for the wonderful introduction on biofilms. By way of declarations, we are developing some antimicrobials targeting RLA, which I'm going to be talking about today for clinical application, and formed a paper Tiger Company, ASRI Bio, towards that end. Okay, so Lauren has partially touched on this. Why are biofilm bacteria recalcitrant to antibiotics? I think everybody knows this. I remember what really got me into this field was reading a news piece that was published in Science in the fall of 1996, and there were some quotes in there from Bill Costerton, who had just hosted the first ASM biofilm meeting in Snowbird, Utah. And he commented into the reporter that the bacteria in a biofilm could be resistant to 1,000 times the concentration that would kill the planktonic form. And we had been studying otitis media, chronic otitis media versus acute otitis media, just like Lauren, for a number of years at this point, and it was like kind of like this light bulb went off in my head. I go, oh, bacteria must be biofilms. So with that in mind, biofilms are heterogeneous, okay? And heterogeneity in a population, whether it's phenotypic or genotypic, helps to ensure the survival of the population as a whole. I mean, this is the universal, across all domains of life. And biofilms are highly heterogeneous. In the case of biofilms, each cell, depending on its metabolic state, its position in the biofilm, and other environmental factors, will respond differently to antibiotic challenge. And our antibiotics were developed and designed to kill rapidly metabolizing and dividing bacteria. So, how do antibiotics function? Well, all extant classes of antibiotics attack what we term macromolecular processes, including protein biosynthesis, cell wall biosynthesis, DNA replication, RNA synthesis. But why would interfering with these processes kill the cell? Well, it comes down to oxidative stress. And this recognition of this general mechanism by which antibiotics kill really came out of Jim Collins' group at Boston University. He's now at the Broad. But they published a paper showing this, and then multiple follow-up, papers starting like in the mid-aughts. And although I didn't know about that at the time we were starting our work in this area, I didn't believe that the resistance was due to lack of penetration of the biofilm matrix. When I first met Bill Costerton and Phil Stewart, that was the kind of the predominant thought at the time. But I did know about penicillin selections, which was a way that you would generate bacterial auxotrophs. So if you're doing a penicillin selection, you would take and you wanted to get bacteria that were auxotrophic for, say, proline. Okay, so you would treat them with some general mutagen, and then you would provide them, with growth media without proline. Okay, and so all the ones that would, that didn't require proline would grow, and then you'd hit them with antibiotics and you'd kill all the dividing ones. And you go through like 2 or 3 cycles of this. And then to get the proline auxotrophs, after you treated with antibiotics multiple times, you would now add proline and they would come out. They were there. So the ones that weren't metabolizing, we knew, could survive antibiotic treatment. And so working with Phil Stewart at the Center for Biofilm Engineering in the late 90s, we actually demonstrated that the antibiotics do penetrate very rapidly. They're small molecules into the biofilm matrix. The only one that was slightly retarded was vancomycin, which is kind of the largest, of the antibiotics. But still, it wasn't enough, to account for the type of metabolic resistance that they were seeing. And so this kind of supported, my concept that it was due to metabolic quiescence. And then we showed that by limiting oxygen, anaerobiosis provided protection. And using oxygen sensing microelectrodes, we could show that the cells deep in the biofilm actually didn't receive any oxygen. They were expiring very slowly. And these were the ones that survived. And we showed that young biofilms with rapidly dividing cells that grow anaerobically were protected, again suggesting a role for oxygen and antibiotic toxicity. And finally, we showed that if you gave bacteria deep in a biofilm, something that could ferment so it would keep growing, or use as a terminal electron acceptor, we could increase antibiotic killing even in the presence of anaerobiosis. And so this is just a slide showing just how rapidly oxygen decreases as you go into the biofilm. So what you see here on the x-axis is the distance into the biofilm, and so 0 represents the surface of the biofilm, as you can see. And by the time you're in 10 microns, there's already a substantial loss of oxygen pressure. And by the time you're in 40 and 50 microns, it's largely anaerobic. And using green fluorescent protein, this thing's gonna be crazy here. Yeah, okay, finally got rid of it. Which requires oxygen in order to be green. We grew bacterial biofilms, and so you can see as near the surface, there's lots of GFP being expressed as green, but the cells deeper in the biofilm are not able to produce the GFP because there's no oxygen. Now, if you look at a young biofilm, the whole thing is green. And it's just because the cells at the top, use up all the oxygen, and you don't get penetration deep into the biofilm of the oxygen because the ones at the top are too greedy. And then we just showed with pretty much every class of antibiotic that we looked at that there was much greater killing of the cells, the bacterial cells, if they were growing aerobically versus anaerobically. So these studies led us to believe that the resistance was passive. That it was just due to metabolic quiescence. We were wrong. As one of the greatest mistakes I ever made in my life was not recognizing actually this. And I got a paper to read that came from Pradeep Singh's lab, which actually identified that the resistance, the metabolic resistance, was active. And as soon as I saw the paper, I'm like, oh my God, I could have had a V8. Anyhow, what they showed was that it was due to triggering of the stringent response. So I'm going to spend a few slides talking about what the stringent response is. So the stringent response is this ancient and highly conserved mechanism among essentially all bacterial species. And it's triggered either by starvation or stress. And in particular, the type of starvation that's been studied the most is amino acid starvation. Well, depending on the species of bacteria, the class of bacteria, there are other starvation triggers that can trigger it as well. But what the stringent response does is it turns a bacteria into the bacterial equivalent of a stem cell. We call them persisters. So if you hear the term persister cell, that's essentially the bacterial equivalent of a stem cell. And stem cells, whether they're bacterial or human, are characterized by a switch in metabolism from growth and division to long-term survival mode. And survival is ensured by upregulation of mechanisms to combat what? Oxidative stress. Now remember, we just showed that oxidative stress is what kills when you treat with antibiotics. But if the cells have already upregulated all their machinery to deal with oxidative stress, then they'll survive when you treat them with antibiotics. They upregulate their superoxide dismutases, stress response genes associated with protein integrity, glutathione production, again, which protects against oxidative stress. So all of these, metabolic changes allow the bacteria to persist in the face of antibiotics, in part because they are able to deal with the oxidative stress that's produced by the antibiotic treatment. So, of course, this immediately suggested a target for the killing of biofilms, REL-A, which stands for relaxed A. REL-A is a protein that sits on the ribosome. It's kind of part of the ribosome, if you will, and it senses starvation, particularly amino acid starvation. And it produces a special type of a hormone or a second messenger called an alarmone that triggers this wholesale switch in metabolism from growing cell to stem cell or persister cell. Thus, we reasoned if we could develop a drug to inhibit the action of REL-A, we could repotentiate all antibiotics against biofilm bacteria, is they would not be able to upregulate their oxidative stress response mechanisms. So when I first got to Drexel University, which was just after the publication of the paper from the Singh lab, I knew I needed to find a collaborator or collaborators to work with who were organic chemists. So I went over to the chemistry department and gave a seminar on this topic, and Frank G, hyphen G here, said it sounded like a great project. He had just been recruited from Oak Ridge National Labs. And so I didn't hear anything from Frank for about a year. And then about a year later, he said, I found a graduate student who wants to work on that project. And that graduate student is DJ Hall, who is now my close collaborator here. He did his PhD with us, and 19 months later, he was made a faculty member for the extraordinary contributions that he has made to science in a very short amount of time. He was also, in addition to being mentored by myself and Frank, mentored by my former lab chief, Dr. Yara Kroll, and Felicity Tso is a graduate student who's now working with DJ and me on this project. So what does REL-A do? Okay, REL-A binds both ATP and either GTP or GDP, okay? And what it does is it transfers the pyrophosphate from ATP to either the GDP or the GTP, forming tetra or penta phosphate of guanine. So you end up with either, with three phosphates, on the 3' and on the 5' and two on the 3' hydroxyl. And so one of the first things that DJ did was to look at a number of various different REL-A proteins and show that there was this enormous structural homology. We're actually very fortunate because just prior to DJ's joining the lab, for the first time, people were actually able to get high quality structures of REL-A and related proteins from this RSH family, which is the REL-A-SPOT homolog superfamily. And the reason for this was that REL-A is a very floppy protein, if you will, and no one had ever been able to crystallize it. And so with the technical revolution wrought by being able to do cryo-EM structures, Brown et al. Published a high resolution structure of REL-A, and very shortly thereafter, there were multiple other structures from the RSH family published. And so DJ overlaid these and could show, that they were all very highly conserved, and that there were two tyrosines denoted by these blue arrows here at 310 and 319, which were extremely highly conserved, and he reasoned that they were probably, because they were both in the active site, probably very important for the function of the REL-A protein. And so he knocked out both the 319 and the 310 tyrosines. And you can see in this slide here that it completely abrogated the ability of the mutant REL-A's to produce magic spot. So the NC in both cases is the negative control, the PC is the positive control, and then the mutants that he made. You see two different spots here on these because you get both the penta and tetraphosphates made if the REL-A is active. So then what DJ did is he took the structure and he built this grid around the active site, okay, which allowed him to do molecular modeling. Basically, the idea is now that we knew for sure that what was thought to be the active site was the active site through his mutagenesis work with the tyrosines, then you could really focus in on looking at how small molecules would interact with this active site. And so he ran about 4 million compounds in silico through this quantitative chemistry pipeline that he developed using the Schrodinger system. And so this slide here kind of shows basically it's a series of ever increasing resolution filters. And so the first filter looks fairly crudely at how a small molecule would bind within the active site, but it's fast. So it makes about 2 seconds per compound. Then you take like the top 15% of those that look like they bind well here, and you look more carefully with the next filter, and this takes about 10 seconds per compound. And then you take the top 10% of that and put it through the third filter, and this takes a lot longer, but it's looking much more carefully now. It takes about 2 minutes per compound for here. So you can see you can't just start here, because if you're trying to screen millions of compounds at 2 minutes a compound, it would take forever to go through. So that's why we use this kind of funnel effect, if you will, for the screening of the compounds. And so out of this screen of millions of compounds, DJ identified at least 40 that had predicted, to bind tightly, within the active site. And the two that are in green were ones that were, or excuse me, in yellow here, C14, which we later renamed Hedge 14, and C22 were the ones that actually, in our semi-high throughput in vitro assay, actually, really inhibited the action of REL-A. Not surprisingly, you see a number of them enhance activity because we couldn't tell. We knew they bind, but we couldn't tell just by looking at the structure whether it was going to be an agonist or an antagonist. So we did not expect, and we did not find that treatment of bacterial cultures with these compounds would materially affect the growth of the organisms, because all you're doing is inhibiting REL-A, which is only going to come on when they're in stationary phase. So with PaO1 you do see a slight increase in the number of bacterial cells once you start getting to the stationary phase. So the first thing we wanted to do is we said, okay, so this Hedge 14, it looks like, in the in vitro assays, it looks like, it has some properties. Does it have any cytotoxicity? And so we just did some simple assays with the human A549 cell line, and even at the highest concentrations, we didn't see any real toxicity with this at all. So we thought, okay, well, it's at least worth going forward and taking a look at this compound and how it affects the bacteria. And so we also looked at the ability of the compound. Did it in an invertebrate model, did it have any toxicity? And so we used the waxworm model, these Galeria. And the waxworms, if they're healthy, they're white, and if they're dying or dead, they're brown or black. And so what you can see is that treating them with our compound, didn't cause any death any more than than the control DMSO, which was our solvent. So that was good. In this simple invertebrate model, we're not seeing any toxicity. But what were the biological effects on the bacteria? And so we showed that there was actually, substantial, a decrease, in the detection of magic spot, or which is produced by the REL-A, in a number of different bacterial strains. And then Felicity has more recently developed this aptamer model. So it's a magic spot RNA aptamer. It consists of a magic spot binding aptamer, a transducer, and this broccoli fluorogenic RNA reporter. And so when it binds magic spot, the aptamer undergoes a conformational change, which allows the transducer to hybridize and allows the binding of the HIB1IT, and it allows the folding of the broccoli reporter, which then allows for fluorescence. And so basically it's a fluorescent assay for the presence of magic spot, or PPGPP. And so this was just a slide showing here on the right, that in the presence, of, Hedge 14, that you get a lot more magic spot produced than on the left where there's no treatment. And here again, you can see, so this is kind of more quantitative. You can see that the green, which is the Hedge 14, reduces, the amount of magic spot compared to all the controls. But what we really wanted to know was, did it enhance the activity of antibiotics? And so we'll just look like here at the left-hand side. This is Haemophilus influenza RD, kind of the lab rat strain of H flu, growing, untreated, 10 to the ninth bacteria. If you treat with kanamycin, you get, close to a 2 log reduction. But if you treat with kanamycin and Hedge-14, you get a further log reduction. And this is with the planktonic, and in the biofilm form, you get a further 2 log reduction. So this is exactly what we were looking for. This compound is potentiating antibiotics, and this is what we had hoped for. What we did not anticipate was a very serendipitous result, which I'll spend the rest of my time talking about. So it turns out that the magic spot levels, or the GPP levels, are actually under kind of a rheostat control. It's not all or none. I was actually under the belief when we first started this project that you either were not under a stringent response or you were, and it was on or off. Well, it turns out that's actually not the case. There's basal levels of anywhere from 5 to 50 micromolar inside the cell. And then when you get to the stringent response, which is what results in the wholesale change of transcriptional and translational activity, you're at 500 to 1,000 micromolar. But in between, there's what's called this adaptational program at between 50 and 500 micromolar. So this is at lower concentrations than actually trigger the stringent response itself. And it turns out that most bacterial virulence genes are under control of this adaptational program. So what that means is that at lower concentrations of magic spot that are required for the stringent response, you turn off bacterial virulence. So we refer to this as defanging the pathogens. Basically, you're taking a pathogenic organism and turning it into a commensal organism. I've got a couple other slides in here. I'm just showing some of what goes on. So in this case, we're looking at E. coli C, which is the most robust biofilm forming strain of E. coli. So you can see untreated here at the bottom of this huge biofilm formed. And if we treat it with, Hedge-14, it completely obliterates its ability to form biofilms. This is one of the first indications that, that this compound was acting, in a way that we didn't anticipate. And the other compound that we'd also been following reduced the amount of biofilm formation, but didn't obliterate it. And then now we're looking at Pseudomonas aeruginosa, and this is where things really started getting interesting. So Pseudomonas aeruginosa, since culture is usually kind of this greenish blue, and it's because it produces piocyanin, and piocyanin is one of its major virulence factors. But if you look when we treat the culture with the Hedge-14 or C14, it turns it yellow. It's shut off piocyanin production almost completely. And this was recognized very early on, actually, by Yarrick Kroll, as I said, who's DJ's in-lab mentor. And so we then went back to our waxworm model, and we took supernatants from cultures of Pseudomonas aeruginosa. And if we took the supernatant from the cultures and just use that supernatant along with DMSO, which is our carrier for Hedge-14, the supernatant from the PA cultures would kill the vast majority, the waxworms. You can see that they're almost all brown or black here. But if we treated them, the waxworms with Hedge 14, it protected most of them from death by the toxins, that were in the supernatant from the Pseudomonas. As you can see here, most of them are white. This was highly statistically significant. And then we actually took and injected the waxworms with PaO1. And in the case where we didn't treat them with the Hedge 14, again, they pretty much all died here on the left-hand side. But when we treated them with Hedge 14, they almost all survived. So basically, this is now showing in this simple invertebrate model that we can take a very virulent pathogen and essentially shut off most of its virulence. Now, in our lab, we work mostly with gram negatives and don't have a lot of experience with gram positives. And so we started working then with Mark Shurtleff's, one of Mark Shurtleff's proteges. We've lost so many of our great biofilm researchers. Lauren mentioned we recently lost Paul Studley. A couple of years before, we lost Mark Shurtleff in a boating accident on the Yellowstone River. But he had developed these great staph models of biofilm. And Nathan Archer, his protege now, an assistant professor at Johns Hopkins, has carried that forward. And so we started collaborating with Nathan Archer there at Johns Hopkins. I'm going to skip over this slide, I think. And what he showed was that if you took our Staph aureus and you put it like on a blood agar plate, that it produces hemolysis. You can see like all the way down to 8-fold dilutions here. This V8, there's still you're still getting some hemolysis. But if you treat it with Hedge 14, then the hemolysis stops like at the second level. So now we've got a gram positive, another nasty gram positive, that looks like it's producing less virulence factors in the presence of Hedge 14, just like Pseudomonas did. So this was really pretty exciting, that we were able to affect virulence production in both gram positives and gram negatives. And so then Nathan actually took this into his animal model. So these are mice. He's using MRSA USA 300, which is one of the worst pathogens you can imagine. This particular USA 300 is bioluminescent, so you can study it on one of these IBIS systems. But what you can see here is that in a dose-dependent manner, that we greatly reduce the size of the lesion with a single dose of our RELI inhibitor, the Hedge 14. So if you look kind of down here at the bottom where it's fluorescent, you can see it's very brightly fluorescent if there's no treatment. We treat with 125 micromolar, we get like more than a 10-fold reduction in the number of bacteria based on the amount of light being emitted. And at 250 micromolar of the Hedge 14, there's almost no bacteria at all. And so it's this compound as a sole agent, as opposed to being used as to repotentiate antibiotics, is actually more exciting because now we can shut down, virulence production in both really bad gram negatives and really bad gram positives. And so once we got these results, then we felt comfortable in trying to move forward. And we're in the process of raising money for our small company now to be able to start doing preclinical studies and then eventually phase 1 studies. And so that's what I have. Thank you. Happy to answer any questions.
Thank you so much, Garth. That nicely complements Lauren's, right? Because you're talking about the bacterial side while she was talking about post-response. And then While people are putting in their questions, I'll put my question forward for the mentees, because for children that have chronic otitis media with effusion, standard of care would be to give antibiotics depending on the duration of the infection, and then put in ear tubes, which by themselves can also induce biofilm formation, correct? And then for around 5% of individuals with chronic otitis media, especially in children, but more in the adults, they might end up with a tympanoplasty mastoidectomy, where you can remove some of the mucosa or drill the bone if needed, but it will not remove all the biofilms as long as you have remaining mucosa. So I suppose the question is translationally, for both Lauren and Garth, how do you see this? Do you foresee first a preventive mechanism wherein you can have a vaccine that would attack multiple infections, chronic mucosal infections, because the mechanism seems similar, or maybe a local treatment, both from the bacterial side, like H14, and the host side, wherein you have an antibody against the structure of the biofilm, plus antibiotics if needed, or maybe both. How do you see this in the future?
I mean, so obviously, what we've been doing, with the mice, is it's a single, dose, at the time of infection. And we don't know anything about, trying to do, systemic treatment with our compound yet. But I could certainly see, that one of the things we'd actually like to do with it, perhaps in collaboration with Lauren, because she's really got the animal models, is, induce, otitis media in the chinchillas, and put tubes in, and then see, like, if through the tubes, you can treat with Hedge 14, or perhaps a combination of Hedge 14 and Lauren's, monoclonals,
I would agree. I think it's always important to consider the different ways we can combine treatments for the greatest outcome. And you have a very heterogeneous human population. You have heterogeneous biofilms. You have people who have already a standing history of otitis media and existing biofilms, not just otitis media, but other diseases. Everything we can do to both boost the host response and target the bacteria in combination, I think, is where we need to always be thinking. Most of us can't take on everything, right? We have our little niche that we explore. But the beauty of this community is the ability for us to work together to really achieve the best outcome. And I think there's a lot of opportunity here, because these things could very well be complementary.
And I do have another question. So for the biofilms, we've been focusing on pathogens. Do commensals play a role in any of the formation of biofilm, or not at all?
So if you really listen to some of the people who are the leaders in the field, they just don't really see a distinction between commensals and opportunists and pathogens, that it's really a combination of the organism and the host and the conditions that, in otitis media, most of these organisms are not overt pathogens. I mean, the pneumococcus may be the exception, but, they're happily residing in the nasopharynx doing nothing at all. But when they get into a place they don't belong, they just continue to do what they do, which is to try to survive. And we respond in a way that then induces the inflammation, then the disease process starts. So I don't think we think of it so much in terms of, is it a commensal or is it a pathogen? It's more, where are they? And if it's causing disease, then we need to target that and protect them when they're behaving as a good commensal. Just kind of keep them where they belong, because they're serving very important functions. They've evolved in partnership with us. And so we have to be very careful about thinking of it like scorched earth type, approaches, because, when you think about what happens when we got rid of all the capsular types in Prevnar, the other ones just kind of took the place, staph takes its place. It creates an opportunity for others to go in there. So you have to be really careful when you're starting to mess with organisms that do play a predominant and important role with us as commensals, which the otopathogens, if you will, do.
I really like, what you just said, Lauren, because, both, the work that you're doing and the work that we're doing, they're not scorched earth policies, and so I've one of the things that we really want to do, is my hypothesis is because we're not actually killing the organism, although we may aid the host in killing the organism, that there'd be less selective pressure to develop resistance against these, because you're not actually killing the organism, and so I think that's been one of the problems that we've had, and that we've had basically this arms race between humans and antibiotics.
Mm-hmm.
But if we get away from this concept of trying to kill the organism, where you're just trying to stop it from causing disease, I think that's kind of it's almost like a novel, novel paradigm, for thinking about how to treat,
Well, in an immunocompetent host, with most kids with otitis media are immunocompetent, we find that if you just get that bacterial load down, they're very capable of dealing with, once you limit the load from something outrageous to a reasonable number of bacteria. And so I think what Garth is saying and what I'm saying is the same, is that we just need to not over attack these bacteria to the point where we're inducing them to just become more and more resistant and, in a way, pathogenic.
Yeah, when you're giving your talks, I keep thinking back to the talks from Mood Buta and also Peter Santamaria, where they're talking about chronic otitis media pathogenesis, and they talked about persister cells, which could be addressed by betadine or povidone iodine, but it is autotoxic to the ear, and it's still questionable whether that can even be applied to patients. So I think what you're presenting here is a really good alternative, because they're not, at least, have you had any autotoxicity studies regarding the potential treatments that you're working on?
We haven't done formal ototoxicity the way you think about it, but we've looked at whether or not we get penetration into the inner ear, or we see anything that appears behaviorally. We look to see if there's any head rocking or anything indicative that, at least in chinchillas and other animals, and we haven't seen any signs, but that's what we're monitoring in humans now. So I think there's only so much I can do in chinchillas, but really it's a matter of how are we doing in humans. We have looked at dysbiosis though. So we don't want to treat, you put something in the tubes, and we put tubes in our chinchillas, they swallow it. So we wanted to make sure we didn't have dysbiosis of the oral cavity or the gut. And we compared treatment with our antibody or by immunization of the vaccine with treatment with standard of care, which is oral antibiotics, right? Oral antibiotics, we had tremendous dysbiosis in the nasopharynx and the gut with the antibiotics, but we saw nothing. So we published 2 of those studies, and we're now doing a study in non-human primates to determine the same. So hopefully it won't, because you don't want to cure otitis media and give, dysentery.
Right.
Yeah.
Maybe that's a bridge too far, but you know what I mean. Then antibiotics are doing that. We're giving these kids are getting diarrhea and rashes and things because, antibiotics, they just are pretty non-specific.
Yeah.
In many ways.
Any final comments? I don't see any questions or raised hands. If none, well, thank you so much, Lauren and Garth.
Welcome. Thank you.
You're welcome. Thanks for having me.
Yeah, this has been highly informative. We're looking forward to have this as a recording for those who missed you today.
Great. You take care.
Okay.
Thank you,
Okay.
everybody.
Thank you.
Thanks, everybody.